So hi everyone, my name is Pratibha Mishra and I have done my graduation from University of Allahabad, and today I am going to represent my topic which was assigned to me about the proteomics study, the biomarkers that are discovered and have helped in identifying the stages of chronic kidney disorder. So I'll share it. First of all, I would like to explain something about the authors. There were 11 authors. So first author was Zaladdin Makhamajanov from Nazarbayev University, Kazakhstan. So all these authors were from either from medical doctor department, HODs, and nephrology department from KOC Hospital or from Nazarbayev University in Kazakhstan. So their studies were based basically on kidney injury, renal failure, chronic kidney failure, and nephrological studies. Now, like to talk about the summary of the context, the significance of the manuscript. The manuscript, in short, we can say it was basically a research about all the by research question which was taken was that how does different positive and negatively correlated proteins with EGFR levels. EGFR is glomerular filtration rate. In the kidneys, the nephrons are the units of filtration, and then they have a tuft of tissue known as glomerulus. So the filtration that takes place in that and the rate is known as GFR. So EGFR levels, they have been associated with certain proteins. Some are positively correlated with them and some are negatively correlated with them. We'll talk about in the upcoming presentation. So we will just find out what's the relation between these positively and negatively correlated proteins with EGFR level and what is the impact of proteinuria on the CKD patients. Like the study was basically first done only with the help of proteinuria to identify the CKD patients. Later on, it was also based on the EGFR levels and their correlation with positively and negatively correlated proteins. So basically, we had to identify these proteins that act as a biomarker. Like if they are present, then possibility of CKD is more. So methodology. In this study, the scientists have done many methods. Like they have first designed the groups, they have divided the groups, the patient, then their study design was done. And then apart from that, they had done several sample collection, laboratory test, mass spectroscopy. So we'll come to that. The major approach was that the biomarkers were the crucial in identifying and managing the CKD. And second thing was the level of proteinuria. So the biomarkers and the levels of proteinuria and the positive and negative proteins, correlated proteins, they were measured. So these three were the main approaches that were taken. Now let's see how they came to find about positive related proteins and the negatively correlated proteins with EGFR, and second the proteinuria levels. So let's talk about the patients. Patients enrolled in this study design, they were divided into two groups. First group was 88 patients that were positively they were not healthy. They were treated with CKD, and there were 49 controls. That is, they were healthy people and of the same age, because we need to get the age same for comparing two things. We cannot make something very irregular. So make it in a regular basis. We have divided into two groups, and then the participants, they were identified with CKD during nephrology consultations. So we cannot randomly pick anyone and say that that person is having CKD. So obviously, those who were registered at this hospital, medical center, National Scientific Medical Center, NSMC at Astana, Kazakhstan, since authors, all of them were doctors or associate professors from that similar university. So all those patients who came for the treatment and they were diagnosed with the CKD, they were taken into account and they were studied. So 88 patients were taken. And these patients were recruited between March 2022, December 22. Okay, so the age was average age, not the kids, not the adult ones. From 18 to 70-year-old were taken, and they had CKD stage, and three stages are there. So CKD stage 1, CKD stage 2, and CKD stage 3. And then they were categorized by EGFR according to KTIGO 2021 guidelines. And then diagnosis was based on EGFR markers, clinical evaluation based on biopsies. Kidney function was assessed during using serum creatinine based EGFR via the 2021 CKD EPI equation. So there are two guidelines, KDIGO guideline and CKD EPI equation. So I can say in a summary that the first study was done, I think pilot study was done only based on the proteinuria levels, and they were said to be CKD patients. But later on, due to some guidelines, they had to identify some positively correlated proteins and negatively correlated proteins, and then according to their number and the maps and the tables, then they were diagnosed with CKD. So control participants were very healthy and they had no, hypertension or some acute infections or cancer or pregnancy that was not present. While in the patients, there were certain patients who had some infections present or cancer or maybe pregnancy or some people with hypertension, because that could also affect the CKD study. So they were taken in the CKD group. And the control was also with the eGFR of less than 30 ml per minute. Study was approved by the same medical center, National Scientific Medical Center, Kazakhstan. So we were discussing about the methods. In the methods, the second part after the study design comes is lab test. So samples were collected. Two types of sample were collected. First one was blood sample and second one was urine sample. Since in this study, on-spot urine sample were taken earlier. But in this study, they have done certain modifications, like they have kept it for 24 hours and then determined the total urinary protein levels. And the remaining sample was resuspended and stored at minus 80 degrees Celsius for subsequent proteomics analysis. And it was done using acetone precipitation method. While the blood sample, that was also used to determine, like, we go for normal blood test. So all the parameters that we get, all these parameters were defined. Like glucose was identified, lipids, urea, creatinine, uric acid, total protein, and blood count. And this urine sample and blood sample analysis was performed. I can say the instrument which was used, or the machine which was used, was a Cobas Integra 400 plus analyzer at NSMC Kazakhstan. Okay. So the next method is mass spectroscopic analysis. After doing the study design and lab test, the mass spectroscopy came into existence. So first was that the urinary proteins, they were kept overnight 37 degrees Celsius in some chemical, and then trypsin digestion was taken place, and it was the concentration was for trypsin digestion, the concentration was 20 nanogram per microliter. So overnight they underwent the trypsin digestion, then followed by reduction and alkylation. And the peptide mixture were purified, concentrated using a zip tip bag, or you can say zip tip, with 0.6 microliter C18 resin. This was the chemical used for concentration. And after that, they were eluted peptides were processed. They were processed with centrifugal evaporator. These are all the machines or instruments that you can say that we used in this methodology and in the mass spectroscopy analysis. And they were further resuspended in 16 microliter of 0.1% trifluoroacetic acid. And after that, 15.5 microliter of the sample was loaded into liquid chromatography tandem MS, LCMS MS machine. So like we do this loading in gel electrophoresis, they were loaded in the MS machine. So name of certain machines and instruments are also in this flow chart. I have summarized it in the form of a flow chart. I hope that everyone can follow me. So the first one, first block we are going. So there was a Dionex HPLC pump with an Acclaim PepMap 100 C18 pre-column and Acclaim PepMap 100 C18 RSLC column. So this was the instrument used for this mass spectroscopy. So chromatography was performed in this instrument at Thermo-Scientific Waltham, Massachusetts, United States. And then the samples were analyzed using a nanoflow reversed phase C18 LCMS MS instrument. It is also a mass spectroscopy instrument at Bruker Daltonics, Bremen, Germany. With the whole captive spray, ion source was utilized for digesting urinary protein. And so the conditions that were present at that time was obviously a dry temperature of 150 degrees Celsius, dry gas 3 liter per minute, and capillary was 1500 volts. I couldn't mention all the details in one slide, so I'm just explaining it. First scan MS spectra were obtained at 2 Hertz spectral rate, followed by 1 MS spectrum. Then after that, data analysis was also done. 3.4 software was used to analyze the retrieved MS data saved in mascot generic format. This mascot generic format is known as .mgf. So this is like we use .pdf, .jpg, .pnb. So this is a file extension that is used in data analysis, and it was used, this software was used, data analysis 3.4 software. Full scan MS spectra were analyzed in this and then saved in this format. Okay. So proteins and peptides were identified using this software, Mascot 2.6.1 software. I cannot help learn all these softwares, but you have to pay attention to this. So it was in London, UK, against the Swiss Prot database, which was taxonomically restricted to Homo sapiens. So they were analyzing humans, Homo sapiens. So this software was basically for humans only, human proteomes, genomes. So that peptides were identified in this software. Then coming next, the following parameters were applied to the search in the mascot software. Then they were searched, and carbamido methylation of cysteine residues 5 and p less than 0.001. So there's nothing to explain much about this. This is a software in which you have to input your data, your peptides details, and then you have to adjust the, like we do in bioinformatics and many other things, we do data analysis. Similarly, you have to adjust the values, the threshold values, the minimum values required for these protein cleavage was was this 0.05 and less than 0.001. So this were the value that you had to input, and then the oxidation and modification would take place. Then the decoy database search was used for the first discovery rate FDR estimation, and the FDR was set to 1%. Then this is also a setting that you have to do in order to get your database in the mass spectroscopy. Then allowed mass error windows for MS were 100 ppm and zero point 0.0 Dalton respectively. This is the exponentially modified protein abundance index. This is EMPAI, was used for label-free peptide quantification. So after the database, the mass error windows were there in which the data was like the setting was 100 ppm and 0.05 Daltons. And then this EMPAI, we will get to know more about EMPAI later on, that this is the exponentially modified protein abundance index, and this is fixed. Like for a particular protein, EMPI is fixed, and if a protein is more than that, then it is said to be upregulated, and if it is below than that, then it is said to be downregulated. So according to that, we need to adjust it, and then we can go further for that proteome analysis, that okay, it's upregulated or downregulated. Now the CKD chance is more or less, or it is being diagnosed with this medicine, so we have to give this one. Like, it was a biomarker, so biomarker index, you can say it is an index for that particular protein or biomarker. Then the proteomic data set underwent log-to transformation and average and slope-based normalization steps to ensure data consistency and integrity for subsequent analysis. So log-to transformation, this was mathematical calculation, and average and slope-based normalization. We need to normalize the data. In proteomics, we need to normalize the data according to the steps so that we can make sure that the data is consistent. To prevent the outliers, we need to normalize them according to the steps, according to the data set, according to the software which we are using for data analysis, and then we will get data consistency, consistent data. So which is easy for analysis. Like if the data is very irregular, something is more, something is high, something is very different in a different parameter, then it is very difficult to analyze them. But if all of them are adjusted, normalized at a consistent, way, then it is very easy for us to analyze it. So this software helps in this. So this way mass spectroscopy is done. Now the techniques and instrumentation and the new techniques introduced in this study. So after the mass spectroscopy, I just made it like a flow chart or kind of table, you can say. These five things were there: gene ontology, geo-enrichment analysis, and geoanalysis. So this all were done in geoanalysis. First of all, that positively and negatively proteins were identified using the packages in R programming. If you know, in R programming, there are these two packages, org.hs.eg.db and cluster profiler 21. These are the packages in R that were used for gene ontology enrichment analysis. And then two types of analysis was done. Like the first one was correlation analysis, and second one was regression analysis. So correlation analysis was how the proteins are regulated, like positively or negatively regulated, how they affect the EGFR rate. And what is the regression analysis? Regression analysis is a statistical analysis, a way in which between a dependent and an independent variable, we can identify the relation between them, so that in a statistical way. So that is known as regression analysis. So we perform both correlation and regression, and that has a big impact on the result. And the function that was used for enrichment analysis was enrich-geo function. The FDR cutoff was 0.05 was considered. Like in the previous slide, we discussed about the FDR cutoff. It is a kind of threshold value which we have to provide. So it is provided here as 0.05. Then the second is reactome pathway analysis. This was basically just to analyze all those pathways, say immune pathways, homeostasis pathways, and all these pathways, physiological pathways that were associated with the proteins that act as my biomarker in CKD disease. So in a brief way and summarized way, I'm just telling that these pathways were negatively and positively correlated proteins with EGFR. So the pathways that affected EGFR in a positive or negative way were taken place in the analysis. They were there for analysis and adjusted. They were adjusted. Like if it is upregulated, then they were downregulated, and if it is downregulated, then they were upregulated, and identified using a package. In the R language, there was a programming. There is a package, reactome PA package, that is for reactome pathway analysis. So correlation and regression analysis proteins were taken into account, and the function that was used was enriched pathway. Enriched pathways with p value less than 0.05 and adjusted q values less than 0.05. Why it is saying adjusted? Because as I told, that adjusted value, the value which is in present, now you have to modify it, either up regulated or down regulated. Then the next is protein-protein interaction analysis. This was done using string DB version 12. This was a software which we were using, and the adjusted gene identifiers for proteinuria were input into the string web platform. So when we input something in this platform, then obviously will get some output. So the output was based on the analysis. The data that we had input, that was matched and predicted and known data. These two were taken into account and then matched and analyzed. What's the difference between them? What's the interaction between the pathways? What's the interaction between the proteins? How they affect the EGFR levels? And then the data was shown. Then data visualization. Now, after we have known the data sets, we have known their ontology, pathways, and interaction between the proteins. Now we need to visualize that data. So in the form of bar graphs, volcano plots. So the package that was used for data visualization was ggplot2, and heat map was generated, volcano plots were generated, and bar plots were reported, and these were reported in minus log 10 scale for visual clarity. This is a scale like we take for x-axis or y-axis. This centimeter is equal to 1 unit. This particular unit is equal to 1 square. So like this, there is log 10 scale which was used so that the data may show like clarity. Otherwise, it would be too much stuffed, or you can say too much confusing. Too much confusing it would be. And so obviously all these plots and heat maps and all these were generated, and the geoterms and reactome pathways were used for that. All those geo analysis and reactome pathway that we have done earlier, that was taken into account. Now after visualization, we needed to do the statistical analysis. So that was performed using Stata MP280. Normally, as there are always in the statistical analysis, there are positive and negative standard deviations always. So they were positive, negative standard deviation in normally distributed variables, and non-normally distributed variables had median and interquartile range. It is here written as MED and IQR. This is median and interquartile range. And then some certain other tests were performed, and I have highlighted all of them, although it looks much confusing, but I had no other option. So a two-sided t-test and Wilcoxon ransom test, then 3 CKD stage group, then were there. So one way ANOVA and Crystal-Wallace test, a chi-square test, Spearman's correlation test, and then linear regression analysis. As we had discussed, that we will perform correlation analysis as well as linear regression analysis. Then prior presentation, that was like some part of this study was these authors and scientists were working on this. So they had already published some parts of this study at previous sessions that were held at ISNWC 2022 and HUPO 2023 Congress. So there's nothing to learn about. This is the data. This is the fact that they have presented at this place. Then the result, after we did all these reactome pathway, geo analysis, data visualization, protein interaction, and the prior presentation, which was not the method, obviously, then we got the result. The key result, the main result was that if we, these are the part of result, but somewhat say more detailed or somewhat more brief. It's a between. So there were stages 1, 2, 3, and of the same age in both the groups, CKD patients and the control group. And the mean age was 38.6 years. And for patients, and then 36.2 for the control group. The patient group included 48% female and 52% males, while the control group included 69% females and 31% males. Now note this point. This point is going to be a limitation later on because there is a gender imbalance, and that obviously affects the study. You need to have the equal number of males and equal number of females for a particular study. Since the disease was rare, the patients registered, they had to take them into account. So therefore, this gender imbalance was according to the data. Now, metabolic markers including serum urea glucose from blood samples. These data are coming from the blood sample. So what they showed significant variation. That is, P is less than 0.001 or equal to 0.001 respectively. Now, we had done two things: blood sample analysis, urine analysis, and from these two things, we got to know about proteins, positively correlated and negatively correlated proteins, and then proteinuria level was also one of the biomarker for CKD. So proteinuria was higher in patients with more advanced stages of CKD, and then differences in the number of detected proteins and EMPI for total protein between control and patient group. P is equal to 0.007. So earlier I had told that EMPAI is nothing but a protein abundance index. It is the measurement or index that this particular protein is present in this much amount in the body. It should be present in this amount, but it is present in this amount. So the control group has obviously some difference from the patient group, and then there was a big amount of difference in them. So the number of detected proteins was higher in healthy controls compared to the CKD group, and a lower number of protein was observed in patients with more advanced stages of CKD. So what did they note? That in the patients with the stage 3 or can say final stage of CKD, there were less number of proteins and there were higher number of protein in the controls. The healthy people had more number of these detected proteins. Now, if we do it separately, then in the CKD patients and control, then this is the result. And in general, this is the result. Particular proteins, these proteins are negative correlated, these proteins are positively correlated. In CKD patients, these are negatively correlated, these are positively correlated. And among control, these are negatively correlated, these are positively correlated. It might take long if I take name of each and every protein, so I'm skipping that. So, this is data. Clinical and biochemical characteristic of participants, normally distributed numeric variable expressed in plus minus as standard deviation, and non-normally distributed variable as median Q. Earlier slide I had told that in the normally distributed patients variables, the numeric variables were expressed as mean plus minus standard deviation in the form of mean. But in non-normally distributed variable, it was presented in the form of median and interquartile range IQR. Control group, CKD for chronic kidney disease group, and EGFR and EMPI. This is the data. You can look over this and find all the proteins and all the blood related reports, the parameters, serum, cholesterol, creatinine, uric acid, bilirubin, urea, glucose, and then we can analyze that the patients with CKD stage 3 had lesser number of these proteins. Take any one for examples. This one, HGB, hemoglobin. Is it? No. Take ESR. It was 8, 24, 19, and 18.5. So ESR is not good thing, but it was more in CKD patients. And take, for example, serum. Serum protein was more in control group, then lesser in CKD stage 1, more less than CKD stage 2, and then very least in CKD stage 3. Okay? So you can analyze these things, and I'm not going to analyze all of them. So regression analysis shows, we perform, again reminding, correlation analysis and regression analysis. Correlation analysis was given there, and now this regression analysis, also it is performed and divided into 3 categories. I have made it under each block. If we generally look, then the regression analysis, these proteins showed negative correlation, these showed positive correlation. But if we performed it separately in CKD patients and in control groups, then these were negative for CKD patients, positive for CKD patients, these were negative for control groups, and only CRNN exhibited a positive coefficient for correlation with EGFR. So not correlation, it's coefficient here, because this is regression analysis. You can say this term as correlation in earlier because that was correlation analysis. So now, the geo analysis, gene ontology analysis that we had done. This analysis was based on 3 kinds. Like first was biological processes. Depending on that, there were negatively and positively associated things. And then if it was based on cellular components, then it exhibited some features. And then if it was based on molecular function of some proteins, then the negatively associated proteins showed some function and positively showed some function. If we do not go much in detail, but I can just read it out. These negatively associated proteins with EGFR in the biological processes, they had an enrichment in retina homeostasis, tissue homeostasis, anatomical structure homeostasis, and immune response related biological processes. Positively associated, and these proteins had in negative regulation of endopeptidase activity, negative regulation of proteolysis means lysis of proteins, negative regulation of hydrolase activity, all the destructive process, I can say, negative regulation of peptidase activity, humoral immune response, the B cells, then the homeostasis body maintenance system, and the coagulation system process, the blood clot system. Then geoanalysis based on cellular components, like cellular components in the words, you can say microparticle, macrophages, neutrophil, and other things like the components of immune system. Then these cells, immunoglobulins, albumins, these are the cellular components. So based on that study, geo analysis, we got that these exhibited enriched blood microparticle, IgA immunoglobulin immunocomplex was seen, and IgG immunoglobulin immunocomplex only in patients. IgG was seen only in patients, IgG, and IgA was seen to know. And geoanalysis based on molecular function, like what function these proteins perform, what molecular function they are performing. They were negatively, positively associated. So negatively associated protein were enriched in antigen binding, and positively associated protein exhibited enrichment in extracellular matrix, structural constituent, glycosamine glycan binding in patients. This is a heat map that is generated. The x-axis column represents different group, like control group, CKD, stage 1, stage 2, stage 3, and these are all the proteins name. And this is the scale, you can say key, that helps to know that this was like 0 when it is very pale color, then medium was 50, and then when it was very, very severe occurrence, it is present in much more quantity than it is 100. So these proteins that were like very crimson red, these are all present in the range of 100, and all these pale proteins are present in the very low range. So you can identify according to their presence that these are the particular stage 1 group patient, or stage 2 group patients, or stage 3 group patients, or no patient that's a healthy person, control group. Similarly, this is EMPI protein with EGFR in whole cohort. Like what abundance of protein should be present, and then what it is related, what is the relation with EGFR? See, you can refer to this table. These are urinary proteins, and then sperm ratio and p-value, then molecular weights, all these things. Then correlation of MPI with EGFR by two groups. So CKD group or control group. Like in the previous slide, we just compared correlation analysis and regression analysis in three parts. One was in general form, and the second one was CKD group separately, and then control group separately. This same thing is shown in this table. This was for everyone overall. This was specifically separately done for CKD group, and this was specifically done for control group. Types that we had seen, they were gene ontology, results, and this result is pathway analysis result. Now these are the volcano plots. Okay. The red dots are the positive patients, the blue dots are the negative patients, and the black ones are not significant. Proteins, positively related and negatively related proteins. And these are the coefficients. Then they show the relation between EGFR and EMPI of protein. Like what is the presence of this protein and what's the correlation with EGFR and what it should be, like the abundance index should be, that is EMPAI. So this volcano plots help in referring to the data that we obtain. Proteinuria is also one of the biomarker to identify the CKD patient and the control group. If it is present, and this presence shows that there is CKD stage, either 1, 2, or 3. So it is of two types, glomerular, tubular. If it, proteinuria takes place in glomerular area, glomerulus, then that is known as glomerular proteinuria. And if it takes place in the tubular area, PCT, DCT, or any other collecting tubule, then it is known as tubular proteinuria. Then this glomerular proteinuria, if it is in heavy form, then it would cause strain in PTEC, and then that would result in tubulointestinal inflammation and fibrosis. So it is really very harmful, and then it can cause stages to increase and reach to the final stage of CKD. Thereby, it's very, very, although easy to diagnose, but it is very harmful for the patient. And then other diagnostic markers are the kappa and lambda light chain, albumin, and these are the main kappa, lambda light chains and albumin. Or others are either undetectable or are not utilized in diagnostics. Now, what are the limitations or difficulties arising? First is there are a large number of protein, like it's proteomics, and there are lots of proteome data, and so analysis is quite a bit difficult. And high number of albumin, immunoglobulin, and which vary, their concentration varies over orders of 10 of magnitude in concentration. So it's a very, in the form of very small scale, in the form of minus order, and if they vary large number of, you can say magnitude, then it's very difficult to observe them. Then if there are high number of immunoglobulins or albumin, then they can obscure the detection of low concentration of protein. Like if these immunoglobulin was vary with 10 to the power something, 10 to the power that, or this, then they are obviously highly concentration. They have high concentration, and then they can obscure the detection of low concentration protein, which are present in low amount. And if they are present, they can mask the other protein, these highly abundant protein. Okay, so I like to clear two things. First is that if there is high concentration of protein, that doesn't mean that is highly abundant protein. Highly abundant protein is different thing. That is based on MPAI index. That is the abundance index of that protein. And that protein masks the effect of low abundance protein, although it may be highly in concentration. That's what I have learned from this study. Now, and after these analysis and the methodology, then the analysis, we get to know in the final steps, the interaction between the positive and the negative protein with the EGFR were observed. So what was the conclusion? That VTDP, it exhibited the highest interaction among the negatively associated proteins, and CLUS, IC1, KNG1, VTNC, CRU, OSTP, A1, AG2, and UROM, they demonstrated the highest interaction among the positively correlated proteins. So what was the conclusion or the summarized point? That this is the label-free quantitative proteomics, and the sample was taken on the basis of the 24-hour urine proteomics study of the CKD patients and control. Regression analysis and correlation analysis were done. Now, the biomarker was proteinuria and positively and negatively correlated protein with EGFR. Then, underlying mechanism that was involved was also studied. Protein-protein interaction, geo-analysis, the reactome pathway analysis, and data visualization were done. And these all things would identify distinct urinary protein profiles in CKD patients and healthy controls. So negatively associated protein, what was their effect, overall effect? They exhibited functional enrichment in process-related tissue structure homeostasis, as well as in immune system activity. Like we studied cascade pathway and the homeostasis. So the positively associated proteins, they were related to significant enrichment in pathways related to extracellular matrix organism, cellular adhesion coagulation, regulation of immune system and enzymatic activities. Like earlier we studied about the platelets, C protein, and then the protein that causes acute kidney infections, that were seen during this study. Despite the potential, there are implications. Like abundance of that protein index is a semi-quantitative computational approach, and it has inherent limitation and may not represent accurate protein abundance. Then next was validation of these protein using sensitive and specific immunoassays such as ELISA is needed to accurately quantify the protein abundance and confirm the reliability of the results. Then number of participants in this group was limited. So that's the biggest red flag. And then gender imbalance. In the very starting, I had mentioned that there will be an implication related to gender imbalance. For example, males and females should be equal in CKD and control group. Then during urine sample preparation for MS analysis, depletion of high abundant protein was not performed, limiting the identification of low abundant urinary protein in patients compared to controls. And finally, the glomerular disease diagnosis was not validated by kidney biopsy. The kidney biopsies that could not validate this glomerular disease diagnosis, and it can be a research topic as well. This was all in a short. Thank everyone and thank BioPractify team and Vineet Mittal sir for giving me this opportunity to present my views and what I have learned from this research paper. It was very helpful for me. And being a graduation student, this topic was really new for me. Like I had to go thoroughly into this paper like multiple times, but after going through it, I came to learn about many new things and I learned how to present it, how to summarize it in points. And I really thank Dr. Arup Acharji sir for guiding and how to as to present the topics, how to make presentation. I tried my best to reduce the complexity of the presentation, but although since it was research data, I could not avoid certain things, so it was somewhat a bit complicated. So I hope that if there is any mistake or in this presentation, you all can accept my apologies. And thank you so much, Vineet sir. Thank you so much, BioPractify team. Thank you.